So in this class, we will learn cell death. Generally, cell death or loss of cell viability can be defined as irreversible failure of vital cellular functions coupled with structural damage. For a long time, it is generally considered that there are two main forms of cell death, namely apoptosis and necrosis. We mainly talk about apoptosis and then compare these two different forms of cell death. Apoptosis is a form of cell death which occurs as a normal and controlled part of an organism's growth or development, also called programmed cell death. This is a transmission electron micrograph showing apoptosis of human lymphocytes. Light and electron microscopy have identified the various morphological changes that occur during apoptosis. During the early process of apoptosis, cell shrinkage is visible by light microscopy. With cell shrinkage, the cells are smaller in size, the cytoplasm is dense, and the organelles are more tightly packed. Extensive plasma membrane blabbing occurs, followed by separation of cell fragments into apoptotic bodies during a process called budding. Apoptotic bodies consist of cytoplasm with tightly packed organelles, with or without a nuclear fragment. The organelle integrity is still maintained, and all of this is enclosed within an intact plasma membrane. These bodies are subsequently phagocytosed by macrophages, parenchymal cells, or neoplastic cells, and degraded within phagolysosomes. There is essentially no inflammatory reaction associated with the process of apoptosis, nor with the removal of apoptotic cells, because first, apoptotic cells do not release their cellular materials into the surrounding tissue. They are quickly phagocytosed by surrounding cells, thus likely preventing secondary necrosis. The mechanisms of apoptosis are highly complex and sophisticated, involving an energy-dependent cascade of molecular events. To date, research indicates that there are two main apoptotic pathways: the extrinsic or death receptor pathway and the intrinsic or mitochondrial pathway. However, there is no evidence that the two pathways are linked and the molecules in one pathway can influence the other. The extrinsic signaling pathways that initiate apoptosis involve transmembrane receptor-mediated interactions. These involve death receptors that are members of the tumor necrosis factor receptor gene superfamily. Members of the TNF receptor family share similar cysteine-rich extracellular domains and have a cytoplasmic domain of about 80 amino acids called the death domain. This death domain plays a critical role in transmitting the death signal from the cell surface to the intracellular signaling pathways. The intrinsic signaling pathways that initiate apoptosis involve a diverse array of non-receptor mediated stimuli that produce intracellular signals that act directly on targets within the cell and are mitochondrial-initiated events. They induce changes in the inner mitochondrial membrane that results in an opening of the mitochondrial permeability transition pore, loss of the mitochondrial transmembrane potential, and release of two main groups of normally sequestered pro-apoptotic proteins from the intermembrane space into the cytosol. The extrinsic and intrinsic pathways both end at the point of the execution phase of apoptosis. It is the activation of the execution caspases that begins this phase of apoptosis. Execution caspases activate cytoplasmic endonuclease, which degrades nuclear material and proteinases that degrade the nuclear and cytoskeleton proteins. Caspase 3, 6, and 7 function as effector or executioner caspases, cleaving various substrates, results in DNA fragmentation, degradation of cytoskeleton and nuclear proteins, cross-linking of proteins, formation of apoptotic bodies, expression of ligands for phagocytotic cell receptors, and finally uptake by phagocytic cells. Apoptotic cell death is critically important during various developmental processes. Example, both the nervous system and the immune system arise through overproduction of cells. This initial overproduction is then followed by the death of those cells that fail to establish functional synaptic connections or productive antigen specificities, respectively. It is clear that apoptosis has to be tightly regulated since too little or too much cell death may lead to pathology, including developmental defects, autoimmune diseases, neurodegeneration, or cancer. Cancer is an example where the normal mechanisms of cell cycle regulation are dysfunctional, with either an overproliferation of cells and or decreased removal of cells. In fact, suppression of apoptosis during carcinogenesis is thought to play a central role in the development and progression of some cancers. There are a variety of molecular mechanisms that tumor cells use to suppress apoptosis. Next, we talk about necrosis. Necrosis is characterized by cytoplasmic granulation and cellular and organelle swelling and results in the rupture of the plasma membrane and organelle breakdown. Necrotically dying cells initiate a pro-inflammatory response by actively releasing immunomodulatory factors. Here, Figure 1 shows a transmission electron microscope of a necrotic cell. The disruption of plasma membrane and organelles is observable. Figure 2 shows a transmission electron microscopy of an apoptotic and a normal cell. The characteristic chromatin rearrangement appears in A strongly different from its normal organization in N. Compared to the cell in Figure 1, the good preservation of the membrane of an organelle is also evident. Reactive oxygen species and calcium are important mediators of necrosis, but several other mediators have also been described, such as phospholipids, calpins, ceramines, and so on, all of which contribute to the disruption of organelles and the plasma membrane. Loss generated by mitochondria or elsewhere in the cell can cause damage to cellular micromolecules, including DNA, proteins, and lipids. Oxidative damage to DNA leads to hyperactivation of DNA damage response. Proteins suffering from damage by oxidative stress lead to loss of the normal functions and enhance their degradation. Calcium channels in ER and the plasma membrane are also affected by ROS. ROS formation is amplified through several positive feedback loops. All of these ROS-mediated events contribute to the necrotic cell death process. Finally, we summarize the major differences in necrosis and apoptosis. First, from the reasons of cell death, necrosis is always phagocytological, which means cell death happens because of unfavorable stressful situations. For example, infections, excessive heat, radiation, or lack of blood supply. Apoptosis is most commonly physiological. It is programmed cell death which eliminates unwanted cells from the body. Sometimes apoptosis happens in cells with damaged DNA. Second, in terms of cell size. Cell size increases in necrosis, and the cell shrinks in apoptosis. Third, nucleus in necrosis undergo fragmentation and karyolysis. Apoptotic nucleus breaks into nucleosome-sized fragments. Fourth, plasma membrane. There is a loss of plasma membrane integrity in necrosis, but plasma membrane remains intact in apoptosis. Fifth, in necrosis, there is leakage of cellular contents, but not in apoptosis. Lastly, leakage of contents attracts inflammatory cells, so there is inflammation in necrosis. Apoptotic bodies are phagocytosed so that there is no cellular material to induce inflammation, hence there is no inflammation in apoptosis. There is evidence of other forms of non-apoptotic programmed cell death that lead to new insights into cell death programs and reveal their potentially unique roles in development, homeostasis, neoplasia, and degeneration. It has become increasingly apparent that cell death mechanisms induce a highly diverse array of phenotypes and molecular mechanisms. The widespread involvement of different forms of cell death in the development of disease leads itself to therapeutic intervention at many different checkpoints. Understanding the mechanisms of apoptosis and other forms of programmed cell death at the molecular level provides deeper insight into various disease processes and may thus influence therapeutic strategy. If you feel interested in this field, please refer to these literatures for more information. Thanks for your attention. Bye.